Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us, the 123rd 123 seminar, and I call it an European Innovation Council Day. I am especially excited today to have two amazing female speakers. Although female researcher status has been enhanced for the last decades, we have yet to improve more. For example, I have tried to increase the number of underrepresented and female speakers, increasing, for example, percent of female speakers from 21% in 2021 and 22 season to 27% in 2024, but that's not sufficient to me and to the community. I also intentionally made the first seminar schedule, that is 2021, such that the speaker list for the first 4 weeks in 2021 consists of 7 female and 6 male researchers intentionally, including Professor Francis Arnold, Dr. Teresa Good at NSF, Dr. India Hook Barnard at HAR, at EBRC. We should nurture more diversity, equity, and inclusion further. And I want to say one more time, all moms, Wonder Woman, and all the working moms are super, super, super Wonder Woman. So I'm so excited to have two fantastic female speakers today. Okay, it's my tremendous honor to introduce very briefly. It's very challenging, but I will try briefly introduce our pioneer speaker, Dr. Barbara Gerratana. She is a director for European Innovation Council funding programs and SynBYSS executive agency of the European Commission. I met her a long time ago when I was a junior faculty at a conference when she was a program manager at NIH back then. My advisor, Kristala Preder, and other collaborators, and I submitted a center-sized NIH proposal, and I remember that she was very supportive, although we did not get that funding. I was a bit disappointed because it was an exciting project, my first NIH proposal, but I learned a lot from that collaboration and failed proposal submission because we all learn from failure, I believe. Probably more we learn from failure. Importantly, I guess I became a stronger from the failure, as I mentioned earlier, becoming more independent rather than depending on my advisor. So after that one proposal, I never collaborate with my two advisors, Chris Voigt and Kristala Preder. That was the only proposal, as I mentioned, I submitted with my advisors. Barbara, thanks so much for guiding the research community in the US previously and now in the European country, having a huge impact on the scientific progress and community. And the virtual podium is all yours now. And thank you again.
Yeah, thank you very much, Taiselk, and thanks for this initiative. I think it's great. It's also great not only that you're promoting women and underrepresented minorities in this from the SynBio community, but also this international character. I think, as you pointed correctly, personally, I would like to see a growth in Europe of the SynBio community, as I've seen the growth in the 20 years that I spent in the US pushing for SynBio, especially at NIH. So with further ado, I'm just going to tell you what is the program that I'm representing here is all about, and how can you participate in it, and how can you benefit maybe by participating. So let me just get share my screen. Do you see it?
Perfect.
Okay, great. So I'm going to briefly talk about what the European Innovation Council is, and then I'm going to focus especially on the programs of the European Innovation Council, the ESC Pathfinder, because I think this program is the most relevant to the audience of this seminar. And then I will focus on how young researcher going from graduate students to assistant professor can participate and benefit from this program. And then I just give you just 2 slides of a highlight of what we currently fund in the ESC Pathfinder in the area of synthetic biology that personally I think is not enough. And that's also one of my motivation to be here, to spur more international collaboration and more participation from the SynBio community into our program. So what is the European Innovation Council? The European Innovation Council is a flagship innovation program of the European Commission, and it is implemented by the European Innovation Council and SMEs. SMEs are the equivalent of the SBIRs, executive agency called also ESMIA of the European Commission. So the ESMIA, the European Innovation Council, aims to identify, develop, and scale up breakthrough technologies. It's mainly structured in 3 different programs. They're shown here: the Pathfinder, the Transition, and the Accelerator. And the basic idea is to nurture or support teams that have significant scientific data that could enable the development of a technology that has an innovative and disruptive character. And so the idea is that through the Pathfinder, we allow this team, usually our consortium or from different disciplines, to develop the technology up to the proof of concept or the laboratory prototype. And then this laboratory prototype is further developed in the validation and demonstration in the environment through the transition grant, as well as the transition grant supports the business development. So now we are talking about not only developing the science, but also start to think about what kind of business can enable the marketing of this technology or the development of this technology so that it can reach the patient. And then in the transition grant, you could have a consortium of teams, but also a single participant applying to this grant. And then in the accelerator program, this is actually a program that only supports small companies. We call them in Europe SMEs. As I said, in the US, they're called SBIR. And the idea is to take the technology that are coming from the transition program now under probably only as SME, and allow the scale up of the production of this technology and the entrance into the market through grants, and then so up to TRL 8, and then in the last TRL 9, we also provide equity up to $15 million. So the idea is really to guide a technology more than specific investigators from the TRL 0 to the TRL 9. But as I said, I'm going to focus on the Pathfinder because I think this is the most relevant one. We are talking about having basic science data and still maybe developing some of the data, that basic science data that will underpin the development of technology up to a laboratory prototype. And in a nutshell, that's what the Pathfinder is all about. I just said it. And the technology that, so there are maybe 3 characteristics. We want a multidisciplinary research team to be involved. Usually it's international, simply because we are in Europe. It's not so difficult to be international. You just get 3 small countries that are together the size less than Wisconsin, and you're already international. But Katy, as you will see at the end of my talk, there is actually opportunity to be truly international in our program, as well as we want a technology that has a potential to disrupt the market or disrupt care for patients or disrupt society and the environment in a positive way, of course, especially with a focus to Europe. Because this is overall, it's a European program. So within the Pathfinder, there are two different types of calls. One is the Pathfinder Open, and this is a bottom-up call. It's completely agnostic on the field, even more than NSF or NIH calls could be. This is like the field, any kind of proposal can be sent to the open call as far as it is focused on a technology development and the development of the science underpinning that technology. So no limits whatsoever in that sense. And then we have top-down calls, and roughly the partition between the funding is a bit more than 50% for the open, a bit less than 50% for the challenges. So the top-down call are called the challenges, and these calls have the advantage of have the goal to support research and technology development in specific topics. And the other goal is to group the project support under a given call so that they can together overcome challenges in that given area. And so this slide just gives you an idea of the type of funding that we had in the last 3 years. So we are talking about roughly per year, if you sum the open with the challenge, of around 320 million. Success rate for the open is much lower than for the challenges. And I have to say, unfortunately, this year is going to be even much less for the open. We got an unprecedented number of proposals. We are really struggling with this, it's really a lot. So the success rate is probably going to be much less than what is reported here, unfortunately. Oh, give me a second there. So this is just an overview of the call for this year. As I mentioned, the open call, we just received the proposal, so there's no more opportunity to apply for the open call this year, but there will be most likely another call next year around the same time. So usually opens around March. It gives up to 3 million euros, but more can be asked if duly justified. So there's really not a hard ceiling. You really just need to justify your budget request. And the peculiarity, there is an eligibility in terms of the number of partners that are part of this consortium, but I will describe this a bit later on in my talk. For the challenges, it's usually you can ask funding up to 4 million euros, but also in the case of the challenges, you can ask more if duly justified. And the challenges call will open in June and will close in October of this year. So there's still opportunity to apply for the challenges. These are the challenges call of this year. I put in italics the two that I think could be interest to a SynBio researcher. The first one is Solar to X device. So under this very long title, for the SynBio applicants, what there is photosynthetic devices. So if you're thinking about a synthetic biology, based photosynthetic device to capture solar energy, that could be one of the application. I will send links to Thais so you could actually see the presentation of this each given call to know more about the call and see if your idea fits the spirit of the call or not. The other call that could be of interest to SynBio people clearly is nature-inspired alternatives for food packaging. We all know about the mycelium food packaging. I think some of us have even used it. So this does not come as a surprise. So how can a young researcher participate in the Pathfinder project? in different way, depending where you are in your career. If you are a graduate student or a postdoc, you can raise awareness of this program to your professor or head of laboratory. If you have collaborators from different institutions, especially from different countries, you can do the same and think about whether the research that you're doing could actually be very well supported by officially forming a consortium among your collaborators and be funded by the Pathfinder. So you're more like acting as an awareness, a catalyst more than the doer. But then the advantage is that if your PI has indeed form a consortium and is successful in getting a Pathfinder project, you will have a great experience in working for an international interdisciplinary team of consortium. If you're a postdoc, a scientist, or an assistant professor, you could actually be the leader of one of these teams that form the consortium. So the consortium usually it's formed by at least 3 teams from different countries, and I'll talk about the eligibility a bit later on. But you could actually be one of the team leader. We're not necessarily, there's no rule that excludes senior postdoc or scientist to be a team leader. Usually these teams are then organized or are led by a coordinator, and in this case usually the coordinator is more a person with a senior scientist experience or an assistant professor or even more established professor, associate or full professor. But I have projects where assistant professors are the coordinators. In some cases senior scientists are. So hierarchically is not as rigid, if you may. You just have to demonstrate through your curriculum the necessary experience. And so you could be, in this case, if you're a senior scientist or an assistant professor, you could actually be the driving force in putting together such a consortium and submitting the application. And then I said the rewards can be working or leading and or coordinating interdisciplinary consortium. The opportunity, you also receive opportunity to be trained in entrepreneurship. We offer free training to any member of the consortium, from the graduate students to the PIs, in areas of IP, in setting up a company. There is also a specific program for women leadership as well. If you may, not too dissimilar to what the ACS used to offer. I don't know if they still offer it now. I don't know. I attended like 25 years ago. That tells my age. It was a great program. So something akin to that. But overall, the opportunity of being a Pathfinder project is truly an amazing opportunity to work for a truly groundbreaking projects. So just briefly, what are we funding in synthetic biology? We had specific calls. The first call that I actually wrote in engineering living materials, this really asked for SynBYSS researcher to apply. And I was happy that out of the 7 projects that we funded under this call, we got 5 projects in synthetic biology for a total of 28 million euros. These projects now are organized in a portfolio together. And as I said, they are trying to work to overcome the challenges, both, either whether they are technical or in ALSA or in regulatory issues or in go-to-market issues for ELMs in Europe. The portfolio has a strategic plan. If you want, I have the link in the slides, but also there's going to be a symposium. So the 18th of September, the 4th International Conference in Engineering Living Materials will be held in Saarbrücken in Germany. And the morning of the 18th, the projects from the EIC ELMs the projects funded by the EICLM's call will be presenting. So there will be a symposium dedicated to them. Then, of course, we also add other calls, such as the novel routes to green hydrogen production. In this case, there was one project with some SynBio approach. And then carbon dioxide and nitrogen management and valorization. 4 out of 6 of the projects funded under this call have a SynBio approach. There was a completely missed opportunity. I'm still surprised from the 2022 DNA-based digital data storage call. Apparently, none of the projects that are funded under this call truly have a SynBio character. Also, keep in mind that because our projects are multidisciplinary, with few exceptions, it's not that there is a project that is 100% SynBio. You might have AI, modeling, bioprinting, other material scientists involved. So there is a multidisciplinary character to them. Then, so overall, from the challenges, we're funding 40 million euros of projects in SynBio. And through the open call, we're funding roughly 53 million euros. That's why I'm partly here, because I personally would like to see more SynBio funded through our projects, through our programs. And so we fund 18 ongoing projects right now. And the project's duration can vary from 3 up to 5 or 6 years. So, for example, the ELM's challenges are up to 6 years. I'm sorry, but I think I missed a slide. I just noticed, like, where did that go? Apologies. I'm going to finish and I'm going to go back to that slide. So, but most of the projects that we currently fund are in the sector of health, as you can see. We have projects in synthetic morphogenesis, so using synthetic biology to control stem cells differentiation and development to end up to tissues or even small organoids. We have projects in cell therapies. We have projects in RNA technologies, and we have some projects that are in bacterial chassis for terranosis application and the tissue regeneration. In energy and environment, we have 2 projects. One of them, for example, is doing synthetic biology approaches to develop new biopolymers. And then we have a very interesting project in food and agriculture that use swarm robotic chassis. I can't tell you what chassis they are because all the information is confidential, to detect and attack pathogens in agriculture. But anyway, this gives you an idea of what we fund. But just before I go over the time, let me talk about the boring part, which is the rules of eligibility. So to participate in our project, we need to have at least 3 teams, 1 from a member state of the European Union and 2 from associated countries. So this can be, for example, Iceland, Israel, UK, Turkey. But if you're not from either the associated country, and I have a link here where you can see if your country is associated with Horizon Europe and therefore with the EIC Pathfinder or not, you can still have the possibility to participate in EIC Pathfinder. It will be on top of this minimum requirement of the 3 team from the 3 countries, but you can participate like it used to be for an NIH grant. I don't know if it changed. You can, a foreign entity can participate in an NIH grant, but the funding needs to be approved by the evaluators, as we call them in Europe, or reviewers, as you call them in the US. And so in order to be approved, the US entity or the non-associated countries entity needs to prove that they clearly bring a benefit to the consortium, either through outstanding expertise or through a special access to research infrastructure, to access to a special geographical environment or special data. So with that, and apologies for skipping a slide, hopefully it was not too confusing. I thank you very much for your attention, and I will be happy to answer questions later on.
Thank you so much. It's an amazing talk. And then the good thing I noticed was, basically the very similar program between 2 continents, Europe and the US and also America. That is good because the global problem cannot be solved by 1 country or 1 person. That should be global international collaboration. That is the key. And also I also is glad to see the living engineer living material workshop or conference. Salzburgen, I know probably the people organized by Wilfred Weber and those people,
Yeah.
absolutely amazing people. I invited twice. That's how I went to Germany twice, actually 4 times, but 2 of them is in the same place. We have to interact with those people. So absolutely,
Yes.
thank you.
Yeah. So Wilfred and Tom Ellis are both in the ESCLM's project,
Yes,
so they will
absolutely.
be presenting. And I noticed, I talked to Wilfred regarding your seminar series when you invited me.
Yeah, so thank you so much. That's absolutely amazing, informative, information for many people in Europe, also probably potentially in US. Okay, so now the main
And
speaker
Asia
of
too.
today.
It doesn't have to be the US. Asian country too. They can.
Absolutely, yeah, absolutely.
Africa,
So I'm
anywhere.
Asian, so that's kind of baseline because all the Asian people are representing basically. Okay, so now the main speaker of today with a longer introduction. And to be honest with you, I originally prepared 3 times longer introduction, and then I realized I need to give more time for Katy. Katy Galloway is the WM Keck Career Development Professor in Biomedical Engineering and Chemical Engineering at Massachusetts Institute of Technology, short MIT. Her research focuses on elucidating the fundamental principles of integrating synthetic circuitry to derive cellular behaviors. Her lab focuses on developing integrated gene circuits and elucidating the systems level principles that govern complex cellular behaviors. Her team leveraged synthetic biology to transform how we understand cellular transitions and engineer cellular surface. She earned her PhD degree and master's degree in chemical engineering from the California Institute of Technology, Caltech, BS in chemical engineering from University of California, Berkeley, all the top universities you can imagine. She completed her postdoctoral work at the University of Southern California. Her research has been featured in Science, Stem Cell System and Development. She has won multiple fellowships and awards, including the Cellular and Molecular Bioengineering Rising Star, Princeton's CBE Xavier Lecture Award, NIH Maximizing Investigators Research Award, the most competitive NIH grant, NIH F32, Caltech's Everhart Award. She is true rising star, has organized many conferences, and more importantly, a mom of many, many children, I believe 4 children. I always say that, as I mentioned earlier, all moms are Wonder Woman, but all working mom is super Wonder Woman, and she is one of them. I still don't believe she's a still junior faculty because based on her achievement, she should be already full professor. And I urge MIT professor, give her tenure as soon as possible. Otherwise, Katy will be snatched by Caltech or some other big university. Katy, thanks so much for your amazing contribution to the scientific community. And please take it away. It's all yours now. Thank you.
And thank you so much, Tae-seok, for this invitation and for the very kind and generous introduction. I'm really delighted for the opportunity to share the research that I've been working on. We are, so let me see here. I'm just grabbing. You guys can see my slides, I hope.
Absolutely. Beautiful.
Brilliant. Okay. So I've been fascinated with the power of synthetic biology since I started working in the field. Actually, I got my start when I was at UC Riverside with Wilfred Chen, just engineering proteins. and thinking about how do you take things that exist in nature, fuse them together, and get new behaviors. Very, very simple things. And really, we've just begun to scratch the surface of the different biomolecules that are out there and the different cellular systems that we could engineer. So I'm very excited to expand this world and think about how do we co-opt different technologies and then put those into cells. And specifically, I'm interested in how we engineer cells across different timescales and for different fates, everything from at the scale of protein binding for transcription factors, which is what you're seeing here, then manifests as how cells are actually going to behave. Here you see a T cell actually killing a cancer cell. And so how do we go from being able to engineer protein binding to actually engineering cellular behavior, not just for killing, but then also to potentially build tissues, repair tissues. So these are the things that we'd like to be able to do. And to be able to do that the way biology is already doing that, we need to co-opt, the power of these native systems, these fundamental building blocks. And that's running again. So what we'd like to do with synthetic biology is not necessarily build everything from the ground up, although there's a great utility in doing so because we can understand things more clearly. But oftentimes what we want to do is then wrap around our synthetic circuits and then co-opt certain behaviors and then augment the functions of cells. Most notably, I think synthetic biology has seen a lot of progress and even therapeutic value in going to engineer CAR T cells to fight cancer. And we're really interested in, harnessing, insights that are coming out of stem cell biology and how do you actually use cells to then regenerate and repair tissues, and how can synthetic circuits come alongside and direct those processes and ensure they perform safely. So altogether, my lab is really interested in, filling in this diagram of, how do you actually build, and I'm hoping you guys are seeing my cursor. How do you take a genetic controller, so our synthetic tool, and interface it with some native gene network so you can get information out and into your controller and then instructions back such that you can guide cell fate? And so today I'm going to tell you mostly about our work in reprogramming. This is inherently a synthetic process in which you convert one cell identity, a stable cell identity, a somatic cell, into a new cell type. And I'll be telling you about a process called direct conversion, which is a form of reprogramming. And ultimately, what we want to be able to do is understand this process so we can control cell fate. And then we can look and turn to our synthetic biology tools and build controllers so that what we can do is control gene expression well enough that we can actually then control cell fate. The design objective being, if we can control gene expression of our controller, we can control gene expression of the cell. And if you can control gene expression of the cell entirely, you can then control cell fate. Now, we don't need to control every single molecule or every single gene, but there's a special subset, and I'll tell you a lot about that today. The goal being that we'd like to be able to engineer cells for diverse applications, and we want to be able to do this scalably. One of the things that's been a challenge, I think, in synthetic biology translating is making sure that we're at a scale that's relevant for commercialization. And so the first part of the story I'll tell you today is about how we understood this process and how the individual transcription factors, 3 of which are required, how those different transcription factors actually are important for mediated reprogramming and how that allowed us to build better control systems. And then I will tell you a small bit of the story we've been putting together in terms of how the composition of genetic elements and how they're physically arranged actually leads to specific profiles. And that's informing our design on how we build single vectors where you have multiple genes together, which is really an essential feature if you want to have scalability. So let me tell you a little bit about reprogramming. How does reprogramming work and why is it important? What's amazing about reprogramming is it allows you to take common accessible cells from patients, and those patients could be people who need therapies or have some particular disorders. You can take those cells and reprogram them, move them to a new cell fate. And because you're making cells that are specifically affected within a disease, like a motor neuron in ALS, you can use those cells for disease modeling to understand why this particular patient is having symptoms such as death of these cells. Ideally, then, you can understand more about that process and potentially even do therapeutic screening. So we can identify molecules that will actually rescue these cells and then deliver them back as therapeutics. The reason that's important to do via direct conversion versus instead of taking iPSCs, age fibroblasts retain signatures of aging, Whereas if you go through iPSCs or you have young fibroblasts, there's no signatures of aging. As most neurodegenerative diseases actually emerge in aged cells, this is particularly a more faithful way of monitoring and doing disease modeling. Therefore, therapeutic screening would be more accurate in aged cells, which is why we work on direct conversion. Ultimately, hopefully, that allows us to then identify pharmaceuticals that we can deliver back to patients. But in our most ambitious goal, we don't want to just deliver pharmaceuticals, although that's extremely important. There are cases where pharmaceuticals are not going to be enough, and we actually need cell therapies to replace damaged and degenerating tissue. So how would we imagine doing that? Well, most importantly, this has to be safe enough that these cells are going to be stable long-term. Okay, so how do we actually make these cells? Let's look a little bit at this process. So cells, here we have fate 1, fate 2, and we want to reprogram them. So normally what you're doing is you're overexpressing transcription factor. So here's just one axis of transcription factor A, and as you increase expression, you expect that you're going to be able to move it from fate 1 to fate 2. That's simply what's going to happen. And in theory, what's really happened through the revolution of stem cell biology is we've learned that the world of stem cell biology is sort of flat. If you know the transcription factors, which are all mapped out here on this graphic, you can move from different cell types, whether it's in the ectoderm, endoderm, or up at this iPSC point. You can move along and generate different cell types. And so reprogramming happens when you take a skin cell, add in these different Yamanaka factors, you can make an iPSC. And then if you want to do something where you do what we're going to be talking about today, direct conversion, you can take another set of transcription factors and make neurons. And so we end up doing then is we're adding in these genes, normally on retroviruses, potentially lentiviruses, but ultimately increasing the expression of this transcription factor. And then those transcription factors are then having this radiant effect on these gene regulatory networks. You can actually see the transcription factor P53 bouncing around here in the cell. This is a single nucleus. And you can see that they're having, P53 is broadly acting across this nucleus, turning on sets of genes, turning off sets of genes, ultimately changing cell fate. And so if you think about this axis here, this graph we've drawn here, the transfer function we imagine that's here is really a black box. I guess this is a gray box, but this black box process, all of these different binding events are then changing networks, which ultimately change cell fate. And so this is the process we want to study. So we know our input, and then we can measure our output to study it, but there's a lot of details we are not seeing. So we need to choose a really good test bed. The test bed I'm going to be telling you about today is this system where we make HB9 GFP positive motor neurons. This is a transgenic mouse line that allows us to look at the formation of motor neurons with the GFP signal. We add in then transcription factors. It used to be 6. I'll tell you about how we winnowed down this cocktail. What's nice about this system is you can take a somatic cell, fibroblast, and direct it to a new somatic cell. And these are post-mitotic. So you can study the efficiency of the process and other elements just by counting the number of neurons. You can get to an efficiency, which is something you don't have if you were studying iPSCs. So you can see here, this transgenic mouse, you see the spinal cord lighting up because that's marking the HB9 GFP motor neurons in the spinal cord. Here in a dish, we take the non-fluorescent cells, reprogram them, and again, you can see the fluorescence turning on in these cells. This one actually fully converts. It has the morphology that's proper for a motor neuron, whereas this cell is getting stuck. And so the enigma, the thing that I was really struck by, was that one, the efficiency is really low. Very few of these cells are actually reprogramming, and some of them are getting halfway through the process and then get stuck. And so what's interesting about this is we then have an opportunity to ask why. This is sort of a developmental synthetic biology where we can say we're building this transition to study it. And studying these transitions in vivo is really difficult. But additionally, you're looking at a very developmentally specific state in which the cells start at. And so you can't ask generally, how do cells transit? You're asking from a very specific developmental context, how do they transition? So this is a great opportunity to better understand biological systems. And so we started with a hypothesis that maybe the reason this cell isn't reprogramming is because our programs aren't powerful enough. And that ended up being not quite right. Although some of the data I'll show you suggests that we had figured out how to build more powerful programs. And at the time, anyone would have told you that, well, the reason cells reprogram or don't reprogram is epigenetics. And that wasn't entirely wrong, but it wasn't actually very helpful in thinking about how to move through. So what we started thinking about was, it's not sort of a memory issue like you would have with epigenetics, and it's not really a software issue as you would think with synthetic biology. It was ultimately a processing issue. And what I mean by that is the ability to perform and process our synthetic programs was really what was limiting. And so we had this idea, and it was potentially some elite population of cells that was going to reprogram. They were rare. And so we went through a whole process, which I'm going to skip through for brevity, to then figure out how to expand these elite cells. And there's a pretty long paper that you can dig through. But basically, we were able to identify a chemogenetic cocktail that increased reprogramming about 100-fold. And the take home of how we got there, it can be summarized in this way. We identified highly proliferative cells as cells that on a per cell basis are going to reprogram at high rates. We additionally saw that proliferation was insufficient, but you had to add in transcription. So cells that can transcribe at high rates and proliferate at high rates are sort of the Ferraris of cells. They can do a lot of things that normal cells can't do, and they reprogram. They have mature electrophysiological profiles, and they are able to process the demands of transcription factor mediated reprogramming. How do they get these properties? Well, these cells are loaded with enzymes called topoisomerases. These topoisomerases are really important for mitigating the transcriptional tangles and genomic twists that are induced when you have really high rates of transcription and replication. Basically, tangles across the DNA are then resolved by these topoisomerases, allowing cells to sit in this quadrant where they have high transcription rates and high proliferation rates. And if you don't have that processing capacity enabled by these topoisomerases, you collapse into one of these states, and then your reprogramming rate is very poor. And so we think of this as a processing thing. And so we went from our 8-bit graphics where very little was reprogramming to really high rates, which is exciting because now that we actually have high rates of reprogramming, we're actually poised to ask questions we couldn't ask before. And so again, we are then able to take this elite population, expand or induce it, and then we get really frequent reprogramming. And so then we can go back to the question that was sort of lingering in the background when we first started the reprogramming work, which is, okay, how do the levels of transcription factors actually allow cells to reprogram? Before, when we had really low numbers of events, you couldn't actually say that any perturbation in the combination of cell states here was more or less likely to induce reprogramming because the rates were so low. This is sort of like if you're standing in an empty warehouse and one guy named Tom walks in, and that's all that happens, that day. You wouldn't really know if that's more or less likely than anything else. But if you're standing in that same, warehouse and 100 Toms walk in, you're like, wait a second, is there like a Tom convention in town? And so you need enough events to be able to tell whether something is, causal or out of statistically significant, basically. And so by increasing the rate of reprogramming and the number of elite states, we can then ask if there's specific profiles of transcription factors that are then going to give us higher rates of reprogramming. And so essentially, we needed to do two things. We could think about either improving reprogramming or understanding reprogramming. And ultimately, that's what Nat Wong decided to do together. And he turned to this because in terms of thinking, okay, if I want to improve this process, how do I actually get to improve? Like, we've already increased the rates, but we have 6 different viruses, which is a lot. And how can you understand reprogramming if you have 6 different viruses? That's too many to control. And so the first thing he thought really to do was to build a minimal system where he could have better control by reducing the cocktail to fewer numbers of viruses and also fewer number of transcription factors, both of those. So we started with 6 different factors. And then Nat said, well, I'd really like to deliver one virus with 3 factors. Could we get down to this minimal set of motor neuron factors? And at the time, I thought, well, possibly, but you might lose something. In fact, in reprogramming, we're normally thinking about what do you add to increase reprogramming, not what do you take away. But he said, well, even if it's a reduced rate with this smaller cocktail of transcription factors, I'll have better control, so it's worth doing. And so that seemed fine. And this is about a 14-day reprogramming process. We then add in our transcription factors, neurotropic media, and then at 14 days post-infection, we can quantify and understand efficiency just by counting the number of neurons. And so with our 6-factor cocktail, you can see the reprogramming rate was actually kind of low compared to some of our previous. And we thought that was curious. But nicely, though, he saw reproducibly that this minimal cocktail nil, which is just one virus and then another virus with this booster cocktail, gave us really high rates of reprogramming. In fact, now we were getting to about 100%, and that's based on the number of cells that were plated. We got really high rates. And then our 6 factors was actually much weaker. It was a curiosity, though, because previously we had seen higher rates, and we were wondering what was the difference. You can see from the cell stem cell paper that we had rates that were from 3% up to 100%, but the range was extremely broad. And so we thought that potentially it was something in this cocktail that was limiting in some of these runs or beneficial that might be changing these rates. And so Nat went back and said, what could it be? And he eventually identified BRN2 as being this inhibitory species. So you take any cocktail of factors, in the absence of BRN2, its rate is, you can see basically is going to drop as soon as you add in BRN2, suggesting that it was actually inhibitory, which we didn't know at the time. And so Nat said, well, what could it be doing? Why is it actually inhibiting? So we thought maybe it's actually decreasing the proliferation rate. And indeed, for all of these different cocktails, what you can see on this axis of proliferation, which is the total number of hyperproliferative cells, you can see basically every time you add in BRN2, you see a drop in proliferation. And that was important to us because we had known previously that hyperproliferation was really, really important. And if BRN2 is inhibiting it, that could be how it reduces reprogramming. So we actually went back and verified that our new cocktail did show changes in proliferation, and that those hyperproliferative cells from this nil cocktail were actually reprogramming on a per cell basis at higher rates. The 6-factor ones still reprogram at higher rates, but you're making actually fewer of them. So we know that BRN2 is inhibitory. Curiously, we also find that these hyperproliferative cells actually formed more mature looking neurons. So they fully remodel their morphology. You see that there's these tight somas and processes that are extending, whereas our non-hyperproliferative cells, which are getting more with that BRN2 present, are not quite fully reprogrammed, which is interesting. So then the first thing you might think is, well, maybe the difference between these hyperproliferative cells and these non-hyperproliferative cells is just they got different amounts of transcription factors. In fact, I would bet that the hyperproliferative ones, because they're reprogramming better, are going to have really high levels of these transcription factors. So we tagged each of the transcription factors independently and went to ask what happens. And so looking at our cells, the non-hyperproliferative and the hyperproliferative, for NGN2, ISL1, and LHX3, we actually found something very surprising. The non-hyperproliferative cells had higher levels of expression compared to the hyperproliferative for every single factor. And so higher levels didn't explain it, and that was quite remarkable. But we still know these hyperproliferative cells are going to reprogram at 4-fold higher rates than these other ones that have higher levels. So we wanted to then ask, well, within this population, how do the transcription factor levels change? And we knew that we had to then isolate this one population because of the confounder of higher levels here. So what Nat went in and did was he took and did a titration, and he did this in, I think, a fairly clever synthetic biology way. He took these UORFs and then placed them, different numbers of them, and placed them upstream of NGN2, and then a 2A and Ruby. That allowed us then to look at the different levels of NGN2 expression, or to generate them. And then we could control and hold these other ones constant, and then look at how much reprogramming we got. And so we did this in an assay where we basically have, we infect our cells with these constructs. We then measure at 4 days to say, what is this y-axis, like how much NGN2 level is there? And then we measure the outcome at 14 days to look at reprogramming. So that sets our y. And so what he found was for NGN2, you have these, you can generate a range of NGN2 levels, and you see a positive correlation of NGN2 expression and reprogramming, suggesting that NGN2 is promoting, like increasing the levels and promotes reprogramming. But you could imagine that actually increasing the expression of any transgene might increase reprogramming. It's just a sign that cells are more metabolically active or something. So I was very excited when he went in and tagged the other ones, and we started to see what those profiles look like. And again, I was quite relieved when we saw that for ISL1, actually there was no obvious trend. You certainly need, in the absence of ISL1, you get very low reprogramming, but above that, just baseline expression, almost any level of ISL1 worked. Conversely, LHX3, no reprogramming really in its absence, or very, very low. And then as soon as you increase, you start to see this decreasing trend, suggesting that high levels are actually inhibitory. So that was quite exciting. So then we started to think through, okay, well, maybe these ones don't matter and potentially be inhibitory. But for NGN2, is the answer like, do we just need high levels of NGN2 expression, right? So it may be that in hyperproliferative cells or in any cells, just high expression of NGN2 would dictate what cells do. And so we started to ask, how do we set our mind about whether the levels of NGN2 or the hyperproliferative state, like which one's more important? And we had this idea that the non-hyperproliferative cells would actually reprogram at lower rates, even if you controlled for the levels of NGN2 expression, meaning you don't just need high levels of NGN2, you're actually on two entirely different titration curves, depending on your hyperproliferative or non-hyperproliferative state. And so and that's exactly what we saw. So Nat went in, he sorted these cells out based on hyperproliferation. You can see that even for medium or even high levels of NGN2, you get low rates of reprogramming for non-hyperproliferative cells. But even for medium levels of NGN2, you're getting higher reprogramming for cells that are hyperproliferative, which tells us that you don't need just one of these states. You actually need relatively high levels of Ruby, medium, and then the cells also need to proliferate. Okay, so that then helped us think about how do we approach the challenge of being able to reprogram human cells, where proliferation is notably difficult to achieve. So in the human system, we add in 7 different transcription factors. It takes us 35 days to run this assay. So the grad students who work on this are extremely patient people. And then we could add in this cocktail of oncogenes that increases reprogramming. And so we also then added in our single 3-factor cassette. What we saw was that these oncogenes we had identified previously were not really sufficient for driving reprogramming. The morphology on these cells does not look great. And you can kind of see that actually even better here. So what we decided to do was find more oncogenes basically to drive proliferation and ask the question, can we increase proliferation in the presence of these transcription factors? Previously, we'd seen obviously those factors could be inhibitory, but even LNI with these different oncogenes wasn't proliferating that much until we added in MIR-AKT, BCL2, and c-MYC. And then as we expand this population of proliferating cells, we can see that we're actually making a lot more beautiful looking neurons. You can see here, this is actually a cluster of many neurons sending out projections, and we're getting morphologies that look reminiscent of what you'd see from iPSC-derived neurons. But we're doing this in 35 days without having to go through an IPS intermediate. So that's very exciting. So just to summarize what I've told you so far, we have now been able to take and reprogram cells at high rates. We can do this for human cells. In theory, that means we can get closer to doing disease modeling, therapeutic screening, and understanding how these cells are going to behave, and ultimately getting closer to generating therapeutics or pharmaceuticals. But what we'd really like to ask, now that we have enough cells, can these cells actually serve as cellular therapies? What if we inject these cells? Will they actually survive? What will happen? And so that's the question we were finally able to ask because we had enough cells. So we have 250,000 cells approximately per mouse that we would need for an injection. We paired with Tim O'Shea and his student Honor, who are at BU, to set up an experiment to be able to do these types of injections and ask, can these cells survive? Because no one's ever injected motor neurons, directly converted motor neurons in vivo. And the first question, of course, is like, do you even have enough cells? Because that was a huge question, even just a few years ago. And to put that question in perspective, let me tell you what it would take. The assay I'm going to show you in just a minute, it took about 10 million induced motor neurons to run the mouse experiment. In 2011, if we had wanted to run that with the 6 factors, it would have taken us about 700,000 plates of cells and virus. In 2019, that would have been only 1,800 plates, although I don't have an incubator big enough that I can fit, I think, 1,800 plates. So this would have been quite prohibitive. Just the scale of this was, the rates were so low, we couldn't get to these numbers. Now in 2023, we are now at a place where our yield is reliably 100% because of this cocktail minimization, and we only have about 30 plates of cells and virus we need to prepare. For perspective, that means our yield, again, being at 100%, the cost of running these experiments are, in 2011, it would have been $10 million to run this experiment, we estimated. We ran this experiment last summer for about $2,000 in materials. And for those of you who are really interested in the environment, just thinking about not just there's the cost here, but there's also the cost in terms of like just thinking about the plates that we use. The carbon footprint of the 700,000 plates we would have had to use was about 14,000 kilograms, whereas currently we're down to 0.6 kilograms of CO2 just from the plates. That's the equivalent of 2 round-trip flights to Boston to Sydney for that 2011 protocol. And 2023, that's the equivalent of driving 1.5 miles. So we've really reduced the cost, not in terms of money, but also in terms of the impact on the environment for thinking about how we can scale cell-based therapies. So now that we have scale, we can get to the place where we can ask the question, are these cells actually going to survive in vivo? Again, that's a question we couldn't ask in any reliable way before last year. And we were gratified once we, again, set up these experiments, injected, and then 2 weeks later, found that there was actually HB9 GFP positive cells within the striatum, which is in the mouse brain. We chose this ejection site because it's easy to assay, and we were worried the cells might not survive at all, and then locating them would be difficult. So we were gratified to see that. Additionally, we looked at whether the cells could go into a healthy or a stroke model. And we were, again, glad to see that the cells were actually engrafting better within the stroke tissue, which is good because most of the time you think about delivering a cell-based therapy, you're going to have some trauma to the tissue. And the engraftment actually is even better when you have the stroke, suggesting that potentially the space created by these cells is improved. Okay, so I realized one of my slides is out of order. So you're just going to have to see this all at once. So to summarize, we were very excited that we could improve reprogramming and give us high rates. Ultimately, it allowed us to understand reprogramming. Now that we have minimal vectors where we just have single transcription factor, we can actually think about controlling reprogramming. But we were really, really happy when we got to the place where we could actually inject neurons into the brains of mice. Because again, when I started this work about a decade ago, it was simply inconceivable to think about getting to a scale where you would be able to do a cell-based therapy and observe its effect. Okay, so the last little bit I'm going to tell you about is we were able to now understand how these transcription factor levels are going to promote reprogramming. But we'd like to deliver this on a single vector, and we'd like to be able to control the timing and induction of those factors. So how do we do that? And that we turn towards our design of synthetic biology tools. And I'm not going to tell you too much because I think I'm running out of time, but I'm going to provide some context with some of the work that Casey Love did in terms of looking at some of these lenti vectors. And here you have 2 genes that are going to be encoded on this lenti, a TET-inducible element, and then a constitutive PGK. And then you have the RTTA with the MGL downstream. The reason she tagged this downstream vector is because we started realizing there were weird things happening. And by weird, I mean nothing was happening. You take these lentis and they were just not doing well. And so we wanted to know, is it the expression of the RTTA or is it the expression from the Ruby? Like, what's the limiting feature? So she made some virus so she could see both of these elements. And she could see when she made the virus and put it into 293Ts, you got some mosaicism. Yes, you can turn it on in response to docs. But you have cells that are also not activating it. But really, things got sad when we went into our mouse embryonic fibroblasts, which are the primary cells that are difficult to work with. And those cells basically didn't respond to this lentivirus at all. And so our present challenge, oftentimes in synthetic biology, is we have results that look promising in 293Ts. And as soon as you go and translate to a primary cell, you completely lose therapeutic response. And so summarizing, we are all... we're building amazing things that can work, and we know biochemically can work, but then we run into all of the different controls systems that are in place to resist transgene effects. And it really dampens our ability to then do the type of work where we would be able to encode our programs in primary cells for therapies. And so as synthetic biologists, I think we can either be really, discouraged by that, or we can start thinking about really returning to thinking about how do cells actually mitigate these challenges themselves, because we know two genes expressed on the same piece of DNA can be co-expressed. In fact, some whole developmental programs rely on the precise coordination of two genes on the same piece of DNA. And notably, like zebrafish somatogenesis relies on tight transcriptional coordination from HER1 and HER7 encoded from the same chromosome. And so I'm actually going to skip a little bit, or well, let's see here. I will try to wrap this up because I believe we might be coming close to time here. Is that correct? Okay. So to summarize, we started thinking about how could you actually get more precise encoding. So Chris took on this challenge and thinking about how the physical coupling through DNA supercoiling could actually influence how genes are coupled. And I won't spend much time talking about this work other than to say it's published at Cell Reports, and you can go check this out. The main insight that Chris had was that we're modeling transcriptional activation as if there was no penalty for the supercoiled state of DNA. In other words, RNA polymerase or any other enzyme is going to load onto the DNA irrespective of the supercoiling density along that piece of DNA. And this has to be wrong if you think about the physical degree of difficulty for basically unwrapping a double helical polymer. And so he said, actually, what I think needs to happen is there's going to be a regime where RNA polymerase binding is going to be favored, and that's at the underwound state of DNA. And when it's overwound, you're actually going to have a limited binding. And he took some transfer functions or some energy curves, basically, where from in vitro experiments to basically map this function. What emerges from this is then a very simple model, or relatively simple, in that we expect that RNA polymerase binding will be favored in one context where you have underwound DNA, and then will be inhibited in the context where DNA is overwound. What that leads to then is this supercoiling mediated feedback where the activity of one RNA polymerase starts to bias the probability that you'll have transcription at genes that are up or downstream of that activity. And so you can then model that, which is what Chris did through a stochastic model, looking at either the convergent, divergent, or tandem systems, and then looking at how supercoiling is going to propagate across an axis, as you can see here on this. When we turn on an inducible gene, we can ask, how does the activity of the reporter change? This allows us to ask, if we are inducing expression of one gene, how does it affect the other? And what you're going to see on this axis is just the RNA being made. We then turn on the second gene, and what we start to see is that there's this anti-correlated bursting between the two genes, where positive supercoiling being pushed into the other gene is going to restrict the bursting probability of the other gene. So this is an either-or behavior. Conversely, if you look at the divergent, you can see that these are starting to show coordination, because negative supercoiling will accumulate here between these two promoters, and that starts to bias the probability that the other gene will be on positively. And so you start to get then positive bursting between these two genes. But what scared us the most as synthetic biologists was when we looked at the tandem system, because everyone builds things in tandem. And so this downstream gene is initially on, just like that RTTA we had in our DOX-inducible system. And then we go and turn on our upstream gene, just as you might with the DOX element. And suddenly what you're seeing is the upstream gene is driving the downstream gene to silence. And that is exactly reminiscent of what Katy was seeing in the primary cells. And so that inspired Katy to go back and consider how to redesign her system. And so she took the tandem system, which again, we had seen this mosaicism and then actually a total lack of response in the MEFs, flipped it around to make this divergent system where she has the TRE here going in the opposite direction. And what she saw was actually a very beautiful response in the 293Ts, and we could actually restore the response in the MEFs. And here you can actually see this quite nicely. These are the mouse embryonic fibroblasts, and we can actually turn these on when we deliver a lentivirus, which is very exciting. Just to summarize some of the other things we know about supercoiling, we've recently seen that this divergent syntax amplifies a reporter, not just with an RTTA, but generally. These effects of induction and transcriptional coupling are reversible. So you can add in the docs and then remove it, and you can see the effect dampen, and they're very syntax specific. Additionally, this works in iPSCs where you see tandem syntax. You can change the position of your promoter And basically the cassette that's upstream will dominate, and then ultimately allows us to build better all-in-ones. I guess for the synthetic biology crowd that is here, we're increasingly seeing work where people are using this divergent design. People often ask me if they should switch their design, and I will say, if you have something that's working, just keep working with it. But if you don't have something that's working, think about moving to a divergent design. There's actually increasing, I think, work across the field, but especially I've noticed that the Elowitz lab has been using this divergent design for their systems. It works very well for us as well. So supercoiling mediated feedback is, we think, very important for rapidly coupling and tuning transcription. We think it can give us a very diverse set of behaviors, and that's quite exciting. Ultimately, we think it'll be important then for being able to control the levels of transcription factors in our cells, and getting us to a place where we can build these controllers much more reliably so that we can get to our long-term goal, which is understanding these cells as dynamical systems so we can engineer them for gene and cell-based therapies. And with that, I'm just really grateful to my lab. We've had a really wonderful and interesting start. We started in the fall of 2019. Graduate students started in 2020. So we're very much used to the online thing, but we're very happy to be back in person. We're thankful to our collaborators who've been great. I want to especially call out Tim O'Shea, who's at BU, and his student Honor Adewumi, who did the implantation studies, our funding agencies, and thank you to everybody. I appreciate the opportunity to talk to you about our research.
Absolutely amazing talk. You know, when you ask, can you go on, and I want to say something, but I was mesmerized. I cannot say anything. I want to, listen to continuously just forever.
Oh, that's very sweet. Thank you. I will not go on and on forever. I will stop sharing now.
I like the tiger and cats slide the most. It's kind of joke slide I get. And then the last part is to me the best, because I'm not that much mammalian synthetic biologist, but that be probably universally true. Actually, that's one of my proposal, and we funded, but we couldn't deliver beautiful way than what you just did. So that's absolutely amazing work. I love the last part of the talk.
Oh, thank you.
Let me ask Barbara. Barbara, if you're still there, if you wanted to give comment, question, it's all for you. And then after that, I may ask some question.
Sorry, I was muted. I will leave the opportunity to ask questions to the participants.
So actually, I do not see any question from the audience yet. So you can ask question to her.
One, maybe I see.
Oh, I now see. Now I see. The Maya asked question: can you target topoisomerase or helicases to reserve supercoiling if you want to express more than 2 genes in a short space? This is an interesting question.
That is a great question. Right. So there's work previously from the Richard Murray's group and Jim Collins, where they're looking at a toggle switch and showing that the gyrases in bacteria did influence the switch. And there, I think they maybe saw some positive benefit of expressing them. Mammalian topoisomerases are very large and very complex molecules. So the smallest one, top one, is 2.4 kb. So throwing that into the cells, you can get it to express, but it's also catalytically activated by other proteins. So even if you overexpress it, you don't guarantee its activity. So you might have to overexpress other molecules. Additionally, the other enzymes like top2A it's 4.5 kb, and then putting it in, because it's cell cycle regulated, oftentimes actually leads to a lot of problems because it needs to be very specifically regulated. So you could potentially do that. It might work. We've taken the other approach where we try to push on networks that actually will increase the expression from the endogenous cells. And that seems to work better than just a flat overexpression. Again, because we think that these complex machines need to have the support of the native regulation, and simply our synthetic programs are not doing that. So we see a lot of toxicity.
You know what? I'm interested in medical application of synthetic biology for more than a decade, but I stay in bacterial world. And recently I started mammalian synthetic biology 3 or 4 years ago. It's damn hard. I still do not see the promising data. Well, we are seeing some promising data, but we are doing the organoid work. So I would say you are very brave, working on the synthetic mammalian synthetic biology, and then I still see a difference between the prokaryotic and eukaryotic system, even yeast versus the E. coli or the other bacteria.
Yes, it's very challenging. It was interesting. I was hanging out with Xiaojing Gao, who's amazing, and he said, "Oh, you were so brave to do this." And I was like, "Well, to be honest, if I had known it was going to be this hard, I don't know if I would have been so brave." But you just keep working on the problems that are in front of you, and then also seeing the promise. Cell therapy. I mean, like if you think about what CAR T has done for people, you have people who are walking around alive, who would be dead if it was not for some of the work that people have done. Now, we still are in a challenge where we need to think about how to scalably engineer those therapies and make sure that they are commercializable, like industrial scale availability. But it's so compelling to think that this is an amazing challenge. And I feel like, I mean, Tae-seok, you and I are chemical engineers. It's a chemical engineering problem to think about scalability.
Absolutely.
And to do that with a lot of the details of molecular cell biology is hard. But I think I'm very encouraged to see it so far being promising, and we are making progress.
Yeah, you sound like the Crystal Predator. I mean, she always said you are a chemical engineer, you can do anything, right? So
we hope so. We aim to. Of course.
And we need to do so, because even my father suffering from cancer stage 3, so I need to do something for him. Not only for him, but for many people, in the world. So that's why I was, Anyway, Barbara, do you have any question, comment, anything? Otherwise, we may close, because we are now 11:17.
I know. I took up much of the time, but thank you so much.
No. I spent too much time introduce you amazing two speaker today. So that's all my fault. Okay. If you do not have a question, let me double check one more time. There's no other question from.
There's one. They do it on purpose.
Did you observe stochastic gene expression in divergent? Oh my God, this is an interesting question. Or convergent promoter settings? Actually, we also studied that only, but we didn't see the clear difference. How can we experimentally determine the supercoiling states?
Yes, that's a great question. So we want to answer this question. So a couple of things. It says stochastic gene expression. I think they mean leak. Or that's how I'm interpreting this question. We didn't see a lot of leakage, at least from the TET element. It wasn't leaky. You see a slight increase in the basal activity of the MGL, which is from the constitutive promoter when you throw it divergent. But the TET element, actually, we still don't see expression of Ruby. It doesn't look like it's leaking. And so we were surprised about that. Now, I think that might be unique potentially to the lentivirus. We're looking at MEFs. I mean, the 293Ts did the same. I will say our observations are slightly different. They're on plasmids versus integrated into the genome. So that's something to think about. And that's fully articulated in the Cell Reports paper, and a lot of the data we're seeing, at least experimentally, matches. So to determine the supercoiling states in vivo is ridiculously challenging right now. We're trying to do this. Even doing it in vitro, it takes a lot of work and experimental setup to make those measurements. There are a few probes out there where you can make those types of measurements in vivo, but they're not really high affinity. And so the best possible, I think, way of making these measurements is actually asking how can enzymes access the DNA depending on the supercoiled state. So turning on and off transcription and then asking about enzymes accessing the DNA is the best way we think we can actually ask, is it twisted or not twisted? And if it's twisted, the enzyme can't access, but if it is relaxed, it can access better. That's the best way I think we can get to it, because there's so many other confounders of using transcription or probes to measure it. And then having to average, because the probes require you to have millions and millions of cells and you're tracking a dynamic state. It's almost impossible to do.
Got it. So, I sort of lucky because when I was young, when I was postdoc, I tried multiple different configuration of my generic circuit. And then somehow I found something, but without know the details why that working. So I kind of always use that kind of, configuration.
Yes.
But not many people kind of do so because they just going to use whatever configuration, considering everything should work, but that's not the context always matters, but not many people know about that. Your paper clearly showing context matter and the even orientation matters, right?
Yes. Yeah. So I mean, that's the thing is like, if you have something working, don't change it. But if something's not working, it's worthwhile to try.
Absolutely. That's one
Change.
of the best advice. But I would say just why not just starting using the configuration at the beginning in that way, that doesn't have any issues.
It's been working pretty well for us as the default design. That's where we're starting things from whenever we have 2 genes. And again, it works in most cases. There's occasionally things where we are seeing sequence specific effects, normally like very specific things with promoters that we think are causing different interference. The layers of regulation for even transcription of mammalian cells are myriad. And so then you have splicing as well. So it's hard to know which element you're getting tangled up in when you see things that do fail. And I think that's what's slowed people down is I think people had these ideas that the orientations could matter, but how do you detangle the effects from like the different pieces, parts, and then whether it's integrated on a plasmid, et cetera. It's challenging. And
Okay,
I don't think
so
we've entirely figured out.
Sure. So let me ask some challenging question. It's not relevant, not necessarily synthetic biology question, but more deep question. Even though we have been running out of time, I'm fine. The question is why billion years of the evolution end up in that natural configuration?
Well, I mean, there are no genes that actually have one, I mean, or there's no, I mean, it's not every gene has in the genome will have a particular orientation, right?
Huh?
What's interesting is that there are very specific orientations that do undergird expression profiles. And if you break them, you do see loss. And so in some ways, there's a diversity of organization and order that in many cases, especially I think from the bacterial world, we've always had this idea that you could just put genes anywhere and it would work. Mammalian genomes don't work like that.
That's a
And
difficulty I currently facing.
yeah, and this might be part of why, but I think there are many other answers of why too. And this is supercoiling is probably one part of the answer.
One part of the answer. Yes, absolutely. That is my guess. That's a stupid question and beautiful answer, I guess. All right.
No, smart question.
All right. So let's end. So thank you all for joining and staying today. We'll meet again on now already May, but May 2nd, Thursday, the same time, the same Zoom link. We'll have the Professor Wenjun Zhang at UC Berkeley and Dr. Jonathan Kemla at MIT, another at MIT. As usual, the follow-up informal chat will occur without recording. Please stay here. If you are interested in chatting with us, I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks, I'm stopped recording. Thank you. That's wonderful.